My name's Susan Law. I'm the Knowledge Mobilization Lead for Long COVID Web, and delighted to welcome Drs. Liana Falcone and Dr. Doug Fraser to today's session. I'm just going to get started, though, with a couple of administrative things. And first, a land acknowledgement. We wish to acknowledge this land on which the Long COVID Web operates. For thousands of years, it has been the traditional land on the Huron-Wendat, the Seneca, and the Mississaugas of the Credit. Today, this meeting place is still the home to many Indigenous people from across Turtle Island, and we're grateful to have the opportunity to work on this land. Brief reminder of the purpose of these webinars. It's firstly to share evidence-based information, research results relevant for decision makers, researchers, clinicians, patients and families, and the general public. Secondly, to provide ongoing updates, spotlights on emerging evidence and scientific information about the activities of our national network. And thirdly, to contribute to reducing and managing misinformation about long COVID. We do have a Long COVID Web Code of Conduct, which essentially states that we value highly engagement around the science and its interpretation, as well as its application. A commitment to reasoned discourse, as stated in our Code of Conduct, comes with a responsibility for all to ensure that any comments or questions remain within the spirit of positive, constructive, and respectful discussion. So we do welcome any or all questions and comments in this spirit. Excuse me. Cameras and mics are off, but please post your questions at any time in the chat. We will pause for Q&A after each speaker, and we will do our best to review and address as many questions as possible. And these sessions are recorded, and we do have permission from the speakers today, so we will be posting this session online following the webinar. But we always welcome your thoughts about how these sessions could be more helpful to you. So please take a few minutes at the end to complete a very short feedback survey, which we'll post in the webinar. So thanks, everyone. I think we'll get started. We have welcoming first Dr. Liana Falcone, who's a clinician scientist and director of the PCC Research Clinic at the University of Montreal. And she's also the director of the Microbiome and Mucosal Defenses Research Unit, and she's a Canada Research Chair. So welcome, Dr. Falcone. I'll turn it over to you. Thank you for being here today.
Thank you so much, Susan. Thank you for this great introduction and for this invitation to come and share some of our exciting new developments with our Long COVID Web community. So I'm eager to get started. And here we go. All right, so the title of my presentation today is Host-Microbe Interactions at the Intestinal Barrier in Long COVID, Insights into Pathogenesis and New Treatment Targets. And these are my disclosures. And so the objectives today are to discuss the main challenges underlying the care of people with long COVID, to review the hypothesized mechanisms for long COVID pathogenesis, and we're going to spend really most of the time discussing the role of the intestinal microbiota and its derivatives in immune dysregulation and end organ damage associated with long COVID. So many of us are quite familiar with the extent to which the pandemic has tested our resilience. We know that almost 5 million Canadians got COVID-19. Almost 60,000 have died, and there's about 4.6 million that have survived. And we estimate that 19% of adult Canadians with suspected or confirmed COVID have long-term symptoms, and this is possibly an underestimation of that prevalence. So, you know, lots of us are familiar with the definition of long COVID. I think some of the things I'd like to highlight, however, from the 2024 NASM definition is that not only are we speaking about symptoms that can, or complications that can be present for at least 6 months, but we're now calling this an infection-associated chronic condition, suggesting that some individuals may take a very long time or never recover from their complications after a SARS-CoV-2 infection. So we know that this is a very heterogeneous clinical entity, and we are still very much lacking clinical biomarkers and new treatment targets, because right now we're very much focused on symptom-based treatments, and we're using our clinical evaluation to diagnose individuals with post-COVID condition. So clearly, a deeper understanding of the underlying pathophysiology is needed to help us identify biomarkers and new therapeutic targets. So there's been some really interesting studies that have emerged, and some of the main pathophysiological mechanisms that have been brought out include viral persistence, immune dysregulation that can lead to autoimmunity, perturbations of the intestinal microbiota, which we call dysbiosis, and there's been some suggestion of a connection between the gut and the brain. Endothelial inflammation and microclotting, again, neurological inflammation and mitochondrial dysfunction. And these are all really interesting and are likely now more and more filling up this complicated puzzle. And we're searching to find the connections between these various pieces. So the way we approached this was in February 2021, we opened the IRCM post-COVID research clinic. And in this setting, individuals were evaluated clinically, but in the context of a research protocol. And individuals can be enrolled into the protocol at any point in their long COVID trajectory up until 24 months post-infection. But the scheduled visits are 1 month, 3 months, 6 months, 12 months, and 24 months. And we added to that evaluation post COVID-19 vaccination and after COVID reinfection. So during these visits, there's a medical evaluation. Individuals fill out questionnaires and scores. A standardized set of clinical tests are performed, including physiological tests like an echo or spirometry. And there's extensive sample collection for biobanking, including saliva, stool, urine, and blood. And from the blood, we also isolate serum, plasma, and peripheral blood mononuclear cells, which allows us to do some interesting and informative downstream analyses. So at this point where we started doing some of our mechanistic studies, we had enrolled about 200 participants. And this is a little bit what this cohort looked like at this point. So the average age was 49 years, 2/3 were females, almost 80% were French Canadian individuals. 20% were healthcare workers, and the most common comorbidities were psychiatric, diabetes, and hypertension. And you can see, as is the case in I think a lot of cohorts, is that the vast majority here, 82%, had a mild acute COVID-19 infection. So to give you an idea of the prevalence of some of the most common symptoms, and I'm highlighting the ones that are particularly relevant to this presentation, we see, consistent with other studies, lots of fatigue, lots of muscle pain, and neurological manifestations like trouble with memory, concentration, issues with sleep, either hypersomnolence or insomnia, headaches, and brain fog. So we have two mechanistic studies that have been sort of that we've been working on in tandem, and they're now actually at the point of being submitted for publication. This is the study design of each study, and they're very synergistic. So these both studies start with the initial cohort. We focused on individuals who were not hospitalized during the acute infection. We really wanted to evaluate these individuals separate from those who are hospitalized because we feel like some of the underlying pathophysiological mechanisms are distinct. And in both cases, we honed in on individuals who had provided stool samples because of the link we were making between the intestinal microbiota and the immune responses and what was going on in terms of end organ function. The study we're going to focus on today is the one on the right here, where we've looked at individuals with long COVID who are not hospitalized in the acute infection, compared them to pandemic controls. So these are people who never had symptoms consistent with an acute COVID infection and never tested positive for COVID. And from there, we analyzed their intestinal microbiota. We did some analysis in their plasma, but we took a deeper look at the function of their stool. And I will explain this in more detail as we go forward with the presentation. All right, so first, a look at the microbiota in these individuals. So the first thing I'd like to point out is that we have really differences that are based on biological sex. So if we look at the diversity of the microbiota, that's the number of different bacterial species that we're detecting in the stool samples, we see that there is less microbial diversity in stool samples that came from female participants, as opposed to samples from male participants had slightly more diversity. Traditionally, having decreased diversity in your stool is less associated with health. So usually inflammatory complications, let's say, or other comorbidities. The other thing to point out is that the changes in the microbiota at least seem to be sustained up until 12 months. So individual stool samples were studied at their inclusion visit, which in this case was largely between 3 and 6 months post-COVID. And then there was a follow-up evaluation at 12 months post-COVID. And now I can say we've even looked at 24 months, but the data focus up until 12 months. What I'm showing you here on the right with this principal coordinates analysis is that the samples from patients with long COVID have a distinct microbiome composition that is different from pandemic controls. And again, the fact that you see lots of overlap between the pink dots and the red dots further emphasizes the fact that the composition stays stably changed, at least until 12 months post-COVID. And what you see below in the left C analyses are some of the microbial taxa that are significantly present according to the experimental group. So what comes up in blue, for example, is something that really stands out in individuals with long COVID at their 3 to 6 months visit, versus in green is at 12 months. And then the pandemic control, or here the label has COVID-neg, but their pandemic controls are showing up in this coral color. So we noticed when we were looking at the prevalence of different symptoms, and as I kind of already hinted to you earlier on, there seemed to be a lot more neurological symptoms in female participants compared to males. And we really went forward with our analysis separating according to biological sex, because as you already saw, there's really a difference, right, in terms of microbial diversity. And what we see here, and I know this principal coordinate analysis is not so easy to interpret, but if I draw your attention to the table below, what you can see is that the microbial composition is significantly different between patients with severe long COVID. And in this case, the way we define severe, and I'll apologize in advance, you're going to see two types of labelings, either neuro, no neuro, or severe and mild. They're the same group. So for us, severe are individuals who had lots of neurological manifestations versus those who did not. And they tended to be individuals who had slightly milder symptoms, meaning they were functionally less impacted by their symptoms. So the microbial composition is really different in individuals with the severe or neurological manifestations compared to those who do not have neurological manifestations but still have long COVID. And we see also that there's differences between those who those with severe manifestations and pandemic controls, as well as some differences at the 2 time points here in the neurological manifestation group. And if you look to the right, this is what we call a FAMD analysis, and it's indicating the variables that are really driving some of the changes in microbial composition. And these 2 FAMD analyses they go together. It's just that this is the way for you to see better the label, so they're sort of separated in 2. But what you really see stand out is that the neurological manifestations are really what's pulling the microbial composition. It's an important variable. So we see trouble with sleep, trouble with concentration, trouble with memory, confusion, depression, and brain fog, for example. So this is another look at the composition of the intestinal microbiota. This is just at the phylum level, so it's a very sort of high level look at the different bacteria, and it's just to give you a sense, right, of how things are evolving over time. And what we really see is the most change in individuals with neurological symptoms that are severe and at the 12-month time point. And this is characterized by decreased firmicutes, for example, and increased Bacteroideota, and more presence of proteobacteria. These are all changes that are also traditionally associated with an inflammatory state in other studies of the microbiota with other disease conditions. And here, what we see on the right is a look at what are some of the bacterial taxa that are really important biomarkers according to whether or not patients have neurological symptoms or not, and based on their time point. And I know it's not quickly easy to interpret, but I'm going to just point out a few things. So if we look at the individuals at 12 months with severe long COVID with neurological manifestation, we see that they have a lot of a bacterial taxa that we could not identify. So we're actually trying to culture out the stool and figure out what that taxa is. But we also see significantly more of this other taxa over here, Falcone cola. That's why it's red. And we see also more Bacteroides and more allostypes. And then we see that there's really depletion of other taxa like Faecalibacterium, like Lactospiraceae, like Blautia. And although, we have to get to the strain level, some of these taxa are usually associated with better clinical outcomes. So now we see that they're depleted in individuals with severe manifestations. So next, now seeing that there's definitely a perturbation of the microbiota, especially in women with severe neurological symptoms, we had a look at what we call leaky gut. So markers of increased permeability at the intestinal barrier. And that's something that happens when there's inflammation at the intestinal barrier. And what we see is that individuals with long COVID tend to have higher levels of zonulin in their blood, so increased markers of leaky gut. And it tends to be more increased in individuals who have neurological manifestations. And here, when we look at these correlation plots, we see that there's a distinct and significant association between having more zonulin in the plasma and markers of microbial translocation. So bits of bacteria or fungi, so these are also present in the microbiota, that are going from the gut into systemic circulation. So two of such markers that we looked at were LBP, or liposaccharide binding protein, and beta-declucan, which is a fungal molecule. And we see there's a significant positive correlation with LBP and beta-declucan with zonulin, especially at 12 months. So we next want, this is nice, these are like nice associative data that we saw in our patients, but we wanted to see if we can really show causality using an animal model. And we did this by taking germ-free mice, so mice that don't have a microbiota, and giving them stool from patients with long COVID, either with neurological manifestations or not, and then evaluating the mice. So the mice did not get COVID, they merely got the microbiota from patients with long COVID. And the first thing we see is that there is increased intestinal barrier permeability in the mice that got stool from patients with long COVID and neurological manifestations. And that's shown with increased LBP in their serum. But also, if you look at their intestinal barrier, so this is an immunofluorescent image of their intestine. And normally what you want to see, this is Z01, so it's a tight junction, and you want to see nice tight junctions, nice and continuous. So like the green has to look like a donut here. And what you see in the mice that got stool from patients with severe long COVID or neurological manifestations, there's lots of gaps. It's not a continuous green donut. That means there's some permeability there. So we then decided to look at their neurological function. And what we see when we did neurobehavioral tests is that the mice that got stool from patients with neurological manifestations, they were sort of more agitated in what we call an open field test. So they're literally put in a box, the mice, and they were just moving around a lot more and covering more distance. So this is a marker, this is called hyperlocomotion, and is usually a marker of anxiety. When we looked at their brains, we looked for markers of inflammation, and we saw markers of astrocyte activation. So this is GFAP. So you can see there's a lot more green going on in the mice that got stool from patients with neurological manifestations. And we looked at IBA1, which is a marker of glial cell activation. And what we see is also more IBA1 staining. So this is, again, and I'm showing you this now quantitatively when we actually go and quantify this fluorescence. And it's significantly different between mice who got stool from patients with neurological symptoms versus those who did not have neurological symptoms. So our next question, and I know time's flying, but it was, how does intestinal dysbiosis cause leaky gut and end organ damage in individuals with long COVID? And here, by end organ damage, we really mean like neuroinflammation. So that takes us to something that we find super, super, super interesting. And this is the fact that bacteria make vesicles. So there's sort of these blebs that come off of the bacteria. They're like bits of bacteria, but the bacteria is not dead. And these blebs are tiny, and they're produced by all sorts of bacteria. And they contain a cargo. So these bits of bacteria that bleb off, they're containing bacterial DNA, RNA, and proteins, and they have also like special lipids in their membrane. And they're small, and they can cross the intestinal barrier and circulate into the blood. And with that, that means they can get to lots of other cell types from the host, right? And organs, including the brain. So we really wanted to investigate this further, because we felt that these vesicles, which can clearly interact with host cells, like there's evidence of this, can be having an immune modulating effect, right? They could maybe be causing inflammation and whatnot. So we isolated these extracellular vesicles from all of the bacteria in the stool. So we just take them from the stool of all of our patients that were evaluated. And this is just evidence that I'm showing you, yes, we really isolated the vesicles. This is what they look like. This is the difference in their size distribution, their mode distribution, their concentration, and their protein concentration. So very similar among the vesicles that came from the stool from the different experimental groups that we looked at, namely pandemic controls and patients with long COVID with and without neurological symptoms. So the first thing is we evaluate the function of these vesicles in vitro. So we co-cultured them with intestinal organoid cells. So these are human cells from the intestine, and we grow them in a layer, and we put the vesicles on these layers, and we look. And what we see is that the vesicles that come from patients with long COVID, especially if they had severe or neurological manifestations, these vesicles were much more pro-inflammatory to the intestinal epithelial cells. And we also looked at the resistance across a barrier of intestinal cells when we exposed them to these extracellular vesicles. And normally, if your barrier is nice and tight, then there should be higher resistance, right? And if it's like porous or leaky, then the resistance is lower. And what we see is the vesicles that came from patients with long COVID and severe and neurological manifestations, they induced a much more permeable intestinal barrier. So we did the same type of experiment, but this time using human macrophages. This is a cell line called THP1. And again, we saw the same idea. The vesicles from patients with severe neurological long COVID induced a lot of inflammation. And this is also appreciated in this correlation chart, where we see lots more of these positive and correlations with pro-inflammatory markers at the protein level when there was co-culture with extracellular vesicles from the stool of patients with long COVID and neurological manifestations. We did the same type of co-culture experiments with human brain cells, so glial cells. And again, we clearly saw, and we looked at the gene expression profile of these glial cells, and we clearly saw that the vesicles from the stool of patients with long COVID definitely induced a distinct gene expression signature. And it was characterized by lots of markers that were suggestive of almost like a activated neutrophil kind of profile. And we see this on the gene expression. We confirmed this doing it by PCR, so looking again at individual gene expression, but also at the protein level, we see that there is definitely increased markers of inflammation when there's co-culture with stool vesicles from patients with long COVID. So finally, the last part of this data is we had to prove that this is what's going on in mice. So we took conventional mice. So these are mice with a microbiota. They're not germ-free. And we just gave them just the EV preparation, the extracellular vesicle preparation that came from the stool of various patients. We evaluate one at a time, a patient with severe long COVID and neural symptoms, a patient with mild long COVID without neural symptoms, and controls. And And we gave them these EVs for like 6 weeks. And then we did neurological tests and looked at their tissue. So the first thing I want to show you is that the EVs really do cross the intestinal barrier and go to end organs. And the way we did that is we took EVs and we labeled them fluorescently. And then we basically put them under like a microscope in essence, or as a special imager. And you can see here the yellow is where you see the tagged EVs. So they're definitely like in the gut of the mice. But then if we go and we take out their organs, we can see that there's EVs definitely that are making it to the brain, to the liver, to the spleen and the kidney, but there's a lot in the brain. So we then now had our mice. We looked at their colon length, and if a colon is inflamed, it tends to get edema, so lots of like fluid accumulation, and so it will look fatter and shorter. And so here when we see that the colon length is decreased in the mice that got EVs from patients with severe neurological long COVID. And we also see more LBP in their blood as well. So that's again a marker of leaky gut. And we looked at the cells in their intestine, and we see that again, they're more inflamed in the mice. And if you remember the good old donuts, so the Z01 fluorescent staining, when we look at the ones from the mice who got vehicles, they just got basically like water, like PBS. We see that they have nice tight junctions that are continuous. When we look at those who got vesicles from pandemic controls, they're still pretty continuous. Those from patients with mild long COVID, no neurological manifestations, we see a little bit of interruption, but not much. But we see a lot of interruption and very poor staining in the mice that got vesicles from patients with severe long COVID neurological manifestations. And we looked at their behavior. And I know this is a lot of graphs, but I'll summarize it to say that the mice that got the vesicles from individuals with severe long COVID with neurological manifestations, they had again that hyperlocomotion type of phenotype, but we also looked at their memory. And they, by using something what we call a Y maze, and they did a lot of errors, like going back into the same arm of the maze that they had already seen. So these are like unsuccessful alternations or direct revisits. So we see that there's also trouble with memory in these mice. And of course, we looked at their brain. And again, you can see it's very obvious where we see all the green, the mice that got the vesicles from patients with severe long COVID with neurological manifestations. There's lots of GFAP staining, so astrocyte activation. And there's also more IBA1 staining, so more glial cell activation. So in summary, what we think is going on is we think that with the SARS-CoV-2 infection, there's a perturbation of the microbiota. That's been documented before in the literature. These changes are sustained. That's all in patients with long COVID. That's been documented, but more so in patients who are hospitalized in their acute infection. We're showing that these changes occur also in those who are not hospitalized in the acute infection. They are sex-specific, so they're more pronounced in female participants. And they are sustained at least for 12 months. And this perturbation of the microbiota, which is more pronounced in individuals who have neurological manifestations, well, this microbiota makes extracellular vesicles that can translocate from the gut to systemic circulation and cause inflammation in the brain, which likely is contributing to some of the long COVID symptoms, especially the neurological ones that we're seeing in our patients. So long COVID is clinically and biologically heterogeneous. We need diagnostic and prognostic biomarkers and new treatment targets. Severe long COVID neurological symptoms is associated with dysbiosis, increased intestinal barrier permeability, or leaky gut. And we think that the extracellular vesicles are the effector molecules that are contributing to some of these neurological symptoms that we're seeing. So we made a connection between the gut and the brain that's beyond what we usually see in the literature, which is usually stuff like metabolites that are circulating. Now we're showing that it's the extracellular vesicles that are definitely at least an important player in what's going on here mechanistically. So if you're wondering, what about a potential treatment target? Well, I would start by trying to heal the intestinal gut barrier. It might not be the only part of the treatment, but certainly part of it. It's probably something that we have to do while we're doing something else. And I'd like to acknowledge all the members of my fabulous team who do all the heavy lifting. Of course, a special thanks to our patients, participants, patient partners, and the agencies that fund our work. And this work was largely funded by CIHR. And of course, I'd deeply like to thank all our collaborators across the country and some international as well.
Terrific. Thank you so much, Dr. Falcone. That's terrific. We have just a few minutes for some questions, so I'll just try to rapid fire some of them. So the first one is just around your comment about why or why not, because I thought it had, your statement was that it was considered a chronic illness, but the question is why PCC is not considered an IACC.
No, I think it is.
Okay.
I think it is. Sorry if that wasn't clear. I wanted to highlight that new change in the definition that it is considered a chronic illness. I mean, there's some individuals that recover, but there is a proportion that do not, and therefore now they have a chronic illness.
Okay, thanks for that clarification. So another question, if viral persistence could drive long COVID, how come antivirals don't work?
Right. So we are doing work on this at the same time. And we do see that there is evidence of persistent viral particles. I'm not saying that it's replicating virus in what we've seen, but we are seeing viral particles in patients even 24 months post-infection. And we figured out a link between these viral particles, the inflammation, and what's happening in the gut. My suspicion of why we're not getting to them effectively with some of the antivirals that have been evaluated is because they're hiding, the replicating virus must be hiding in a reservoir and is maybe more or less active in that point, right? It's probably like dormant in a sense. And it's hard to get at. We need some way. It's similar to the difficulties in curing HIV, right? We have a viral infection that's hiding in a reservoir and we can't get to every aspect of the reservoir. Maybe there could be some version of this going on in long COVID. So the approach is somehow you have to get it out, and that's always the challenging part.
Great, thanks. Another question about whether or not prior intestinal damage has anything to do with susceptibility for long COVID.
Yeah, that's a great question and I wish we had samples from patients before they got COVID to really see if we could see a predisposition. I'm going to tell you, in our evaluation of leaky gut in this cohort, there's not an association. It's not like these patients have GI manifestations. I mean, we have some that do, but it's not like you don't need to have evident diarrhea or other, gastrointestinal symptoms to have evidence of leaky gut. And it is my suspicion that leaky gut is probably underlying a lot of things, maybe even other post-infectious conditions or other chronic conditions, especially if there is a link with systemic inflammation, immune dysregulation, and autoimmunity. So I wonder if there are individuals who just have a subclinical version of this and the infection brings it out further. And I say subclinical because maybe they didn't meet criteria, to have the typical positive tests that we do in regular clinical practice, right? But here in the research realm, we have the benefit of going deeper and looking at lots of other biomarkers. And maybe some of these are perturbed in individuals at a sort of, like I said, subclinical level.
That's terrific. Thank you so much. I think we'll turn to Dr. Douglas Fraser now, but perhaps I could invite you, Dr. Falcone, to have a look at the chat and perhaps answer some of the questions just while Dr. Fraser is speaking, because there's several other questions there that we just don't have time for today. But thanks everyone for your questions. They're excellent. That's terrific.
Thank you.
So thank you. And I think we'll turn to Dr. Fraser and your presentation. Thank you. You might be on mute, Dr. Fraser. I can't hear you.
Can you hear me now?
Perfect. Yes, yes.
Okay.
Thank you.
I don't think I can get on camera though. It's not allowing me, but as long as you can see the presentation. So I'll be speaking about two studies. The first is the LC-optimized cohort study, which we started a year and a half ago. And then I'll be talking about a phase 3 clinical drug trial, which we've initiated, which is occurring now in 4 continents. So the first part of our of our work, called LC-Optimize, was an international multi-center cohort study to identify long COVID sub-phenotypes. And the goal was to understand if there were select groups of patients that we could identify and perhaps start to target with therapeutics. We did this in 5 sites: London, Montreal, San Diego, Rio de Janeiro, and Lusaka in Africa. We were able to include close to 6,000 subjects, of which 2,194 had long COVID and 3,367 were long COVID negative. So they had a positive SARS-CoV-2 test, but they resolved their symptoms within 3 months. So they were our comparative group. Just to give you an idea what some of the data looks like, in A, you could see the time frame. So starting at 3 months, when we included people, going all the way out to just over 4 years, and with an age range of about 14 years of age all the way up to 96 or 97. They were fairly equal in terms of males and females, as well as long COVID positive to long COVID negative in terms of age. In B, you can see the most common reported symptoms fell into the neurological system, followed by respiratory, systemic, musculoskeletal, gastrointestinal, cardiovascular, and skin. In C, you could see the symptom occurrence, where fatigue was number 1 across the entire population, followed by dyspnea, myalgia, arthralgia, and so forth. And then in D, you can see the symptoms that occurred when they had their acute COVID-19. The red are the patients who ultimately developed long COVID, while the blue are the patients that did not. And there were some significant differences there. If we take those with long COVID symptoms and we subtract the COVID-19 symptoms, the red shows the new onset symptoms after the infection, and the blue shows the symptoms that occurred during COVID-19 that went away. And arthralgia was in fact the most common new symptom after COVID-19, followed by fatigue and other symptoms, while fever, cough, rhinorrhea, sore throat, diarrhea, etc. all went away more frequently with the COVID-19 or with the long COVID patients.
Sorry to interrupt, Dr. Fraser, but you should be able to turn your camera on now.
Oh, okay.
Sorry, thanks.
No problem at all. There we go. Now, as part of the confusion that's going on with the studies, there were variants of concern, and those variants of concern had not only different timing, but also geographical appearance. So we went to the World Health Organization and other organizations to look at the data of the variants of concern, with wild type being the original Wuhan virus. And then you had alpha, beta, gamma, delta, Omicron. And we looked at the timeframes and the geographical appearances, and we were able to make some associations. So for example, in Canada, wild type and delta and Omicron were the most common, with a little bit of alpha. If you were in South America, it was the gamma and the wild type, which were the most common. If you were in Africa, it was delta, wild type, and beta that were most common. So we were able to make some associations among our study sites. And then we were also able to look at the variants of concern relative to the symptoms. And there were some interesting findings there as well. For example, fatigue was more commonly associated with wild type, alpha, and Omicron, while myalgia was more likely to be associated with gamma, as was weakness and numbness, and gamma was more likely to be in South America. So one can see there's quite a bit of complexity that was going on relative to the variants of concern, the timing, and the geographical areas. We use a lot of machine learning and artificial intelligence in my laboratory, and we were able to look at the data and create symptom clusters. So we were able to make 6 symptom clusters, which was the optimal number, and the features that were most important in placing people in these symptom clusters are shown on the right, with fatigue and dyspnea and myalgia leading. Just to show them a little bit differently, the biggest cluster was cluster 3, which was arthralgia, dizziness, and headache. Then we had other clusters, or I'm sorry, cough and dyspnea. Cluster 4 was the largest. No, it was cluster 5. Again, I'm wrong again as I'm looking at these. Regardless, that they all have slightly different frequencies and different symptoms which are involved. And on the right-hand side, you can see the symptoms relative to the sites and where they are most common. So for example, Lusaka was more likely to be in cluster 3. London and Montreal were more commonly in cluster 4, 1, and 2, and so forth. A tremendous amount of complexity. We then looked at associations again, and the thicker the line, the stronger the association. We have symptom clusters on the far left and in A relative to the symptoms. And you can see that fatigue was in certain clusters, not in others, as was dizziness and so forth. A great deal of complexity. In B, you can see the sites relative to the clusters. Again, there's lots of complexity there. And then in C, you've got the symptoms relative to the sites, and again, lots of complexity. And ultimately, when we put it all together, you get a core diagram that looks something like this. Overall, what our cohort study told us is that there's a tremendous amount of heterogeneity, and it is related to symptom clusters, it's related to geography, it's related to variants of concern, and overall, it's highly unlikely that with clinical data, we're going to be able to identify meaningful sub-phenotypes to target with therapeutics. So our next approach was to move into more of a biological approach. So in the same study, we collected blood from 1,028 patients. We collected blood. You can see the timing from their acute COVID and the male and the females, and they're relatively equal. We took this particular cohort, which is a subset, and when we did our machine learning, we were optimized at 7 cohorts, or 7 clusters, I should say. And you can see the symptoms in B, which led into those, and again, the distribution again. And you see, again, a whole lot of complexity when you look at these different patients. What do we do with that information? We took the blood, we did full proteomics, we did metabolomics, we did viral markers, we did brain injury markers, as well as other investigations. And we've been able in my laboratory to come up with a really, really extensive level of analytics. Our data analysis pipelines are some of the most expansive that you will find anywhere in the world now. We have conventional statistics, we have our artificial intelligence as well, all the way to large language models. When we collected patient data, we also collected clinical narrative notes, so we were able to use large language models to better understand a patient presentation by the actual clinical notes. So we have a really expansive way of looking at all the data. I'm just going to show you the targeted proteomics. We do this now in my lab. We have the ability to measure over 5,400 proteins from less than 10 microliter of plasma. And we can do that in about 1 day. We have all the robotics now to do this. Just to give you some idea, and I know you can't see a lot of this, but just to give you an idea of some of the top proteins which we identified, and there were hundreds, but some are involved in immune function, some in nervous system function, some in cell division, and so forth. When we get a little bit more into the analytics, first of all, we have to do some controls, and we want to see that the medians of the proteins line up in the different groups, which we see on the upper left, that the mean shifts as you would expect. So this all makes perfect sense and looks good. But in the upper right, you'll see the hierarchical clustering, where red tells you proteins have gone up, relatively speaking, and blue tells you proteins have gone down. And you can see healthy control versus LC positive versus LC negative, that there are different patterns. One interesting aspect to come out of our work is that the LC negatives have a proteomic signature, even though they don't report any symptoms. So after having SARS-CoV-2, they have a very, very select proteomic signature. It's not quite as extreme as the LC positive, but it's there. So it begs the question, what's the difference between someone who's reporting symptoms versus someone who says the symptoms have completely resolved? So there's some interesting questions that have come up. When we look at the data as a whole and the proteome as a whole, we had to get into some fairly advanced analytics. In this case, we're using neural networks, and using that, we were able to separate out the patient proteomes quite nicely. If you look on the bottom right, you've got the LC positives, the LC negatives is the two big groups, and down you've got healthy controls down at the bottom. A healthy control for us was somebody who is symptom and disease free, who has not had SARS-CoV-2 in over 1 year or no evidence of such. We can start to deconvolute the data, and one of the things that popped up when we looked at individual cells was that CD4 T cells were dysregulated and in high amounts. This is proportional. You don't see the CD8 cells because they're exhausted after a SARS-CoV-2 infection. And those cells were well represented in the bone marrow, in the brain, and in the lung. We can get into our functional enrichment where we start to understand the upstream regulators and the biological processes. And on the right-hand side, you can see a great deal of ATP and mitochondrial dysfunction, which may be involved in the fatigue process. We get more into global pathway analyses, and we find, again, lots of neurological issues as well as JAK-STAT pathways, which I'll come back to in a minute, as well as complement and coagulation pathways. And then we can start to look at these a little bit closer with enrichment. As well, we look at causal networks, which tells us essentially what kind of communication is going on with these proteins, and certainly platelet activation and neutrophil extracellular trap formation jump out. And these may be instigators of some of the microvascular clotting that's still going on in some patients with long COVID. Ultimately, we were able to determine 13 pathways, despite a tremendous amount of heterogeneity. And if you look at A, you see the symptom cluster in this group relative to the geographical sites, and you see that's very poorly balanced, again, telling you about all the heterogeneity. But despite that, we were able to use our meta-analyses and determine there were 13 pathways which were consistent among all geographical areas and all symptom clusters. In B, you could see the relationships between the different sites and the different pathways that were identified, and in C, you can see the symptom clusters relative to the different pathways that were identified. And again, those pathways were consistent pretty much across the board, which tells us there, despite tremendous clinical heterogeneity and a lot of biological heterogeneity, there's still a lot of commonalities which can be targeted for therapeutic purposes. If we look at the time frame of these pathways and their elevation, so if you look at the dotted line in which is at 0, that's essentially what a healthy control is, and you can look at these pathways and see how they change in the entire population over time. And you see some are improving, like interferon gamma, interferon alpha, reactive oxygen signaling, but then other ones stay consistent or might even be going up, such as TNF and IL-2, IL-6 complement, and coagulation, etc. Some of these are likely to be involved more in the healing process, such as EMT and angiogenesis. But now that we have that data, we want to identify drugs that we could potentially use as therapeutic agents. And ideally, what we want to do is identify drugs that will affect the vast majority of patients anywhere they are in the world, despite their immune priming and despite what variants they saw. So my laboratory uses proteome libraries and drug libraries, and we can link those using our AI pipelines that we've developed, so we can discover new drugs for existing diseases, or we can discover new diseases for existing drugs. And that's just shown below on the left, where we can go unidirectional. But what we've been able to do is come up with pipelines where we can make heads or tails of all this information and start to make logical links. Here are the pathways again, and this is simplified, but it tells you a little bit about what we think is going on, where you have the interferons are elevated, TNF is elevated. Those are activating a number of different pathways, which are then ultimately resulting in platelet activation, dysregulated T cells, reactive oxygen species, coagulation, etc. We've been able to determine that there are 2 drugs which we think will help therapeutically for the vast majority of patients. One is upadacitinib, which is also known as Rinvoq, much easier to say. If you watch CNN, you'll see Rinvoq is advertised regularly. It's a drug that's used in arthritis, psoriasis, and other rheumatological illnesses. And it's a drug that very effectively takes out IL-6 and IL-2 in the middle, but also has intermediate activity against the interferons in TNF. So we believe that this drug will be helpful for many of the patients suffering from long COVID. And this drug could be supplemented with something like clopidogrel, which is a platelet inhibitor, and may also help certain processes as well. Another drug that we've identified through our AI pipelines is pirfenidone. This is used in lung interstitial fibrosis, and it's very effective at knocking out TNF and IL-6 with intermediate activity against the interferons and reactive oxygen species. And again, clopidogrel could be added on to this. So now we've moved into a worldwide clinical trial. We're on 4 continents in 7 countries. It's called LC-Revitalize. It is on clinicaltrials.gov as of today. It's a phase 3 double-blinded, placebo-controlled, multi-arm platform study. What's great about platform trials is they give us a great deal of flexibility under one umbrella. So we're starting off with 4 arms where we have the 2 medications I just told you about with their respective placebos. And then after our enrollment of 384 patients, we'll be doing an interim analysis. And then we have the option of moving forward with 1 drug alone. We can move forward with both drugs. We can drop both drugs, move on to a new drug, or we can do an add-on drug such as the clopidogrel, which is what I was suggesting. In this study, our primary objective is to evaluate the efficacy of the drugs, improving symptom severity in participants with long COVID. We're using the symptom burden.
Sorry, there's somebody named Robert with his audio on. Oh, there we go. It's turned off now. Thank you.
And then we have secondary objectives as well, looking at anxiety, depression, overall well-being. We're looking at safety and so forth. And then we're also doing 2 other things. One is a biomarker evaluation. So we've been able to identify a biomarker panel, which is specific for long COVID. And we've just signed a deal with Thermo Fisher that's creating a multiplex for this use. So we'll be using a specific biomarker multiplex system to be evaluating for improvement. We'll also be looking at the more common markers such as TNF, IL-2, IL-6, which are the pathways which are involved. And then we're funded also to do a comprehensive omics evaluation, which will be proteomics, metabolomics, transcriptomics, as well as whole genome sequencing. So we want to look to make sure the drugs have done what they're supposed to do, and that if there are variants which exist, which will tell us that those people would never respond to those drugs regardless. So this is our overall approach, which we're just starting. So just a quick recap. This is essentially what our goal was coming into this about 1.5 years ago, was to collect patient data, demographic clinical data, to get biological samples. We were hoping that the clinical data would be useful, but it was just so much heterogeneity. It was very difficult to use for therapeutic uses. So we've moved to the biological sample, the omics, some advanced analytics, the drugs in the clinical trials. We've been able to look at 5,561 patients from 5 sites in 3 continents, do blood work on 1,028, do proteomics, metabolomics, viral markers, and brain injury markers as well. We've been able to identify what we believe to be our important pathways, which are consistent in all geographical areas, regardless of the variants involved. We've been able to identify 2 medications, which we feel will be helpful for the vast majority of patients with long COVID. And now we're just starting, and we'll be enrolling our first patients next month in a phase 3 platform clinical drug trial, which will be present at 7 sites in 4 continents. So with that, thank you very much, and I'll stop there.
Fantastic. So a bit of work underway. So congratulations on all of that. We have a few questions here. So the first one is, I don't see the kynurenine. I'm sorry, I'm not pronouncing that correctly. But this pathway, have you checked it just on the side? Are you open to coming to Germany and Hungary for clinical trials?
So in terms of the pathways, we didn't look for specific pathways. It was exploratory. So the pathways were ranked on their relevance and their association. So the pathway may be there, it may be active. It just didn't come up high in our particular analyses. Um, anytime you do a study like this, the biggest problem is the cost. So with everything I've shown you, we're $14 million in. And I just don't know if we would have the money to add any other sites on.
Okay, thanks. There we go. The next one is, what are the primary outcome measures that will be used for the interim analysis? There's a concern there that many current trials do not have outcome measures able to show actual change.
Yeah, so we're using the SBQ, which is the symptom questionnaire, which is a validated questionnaire, which was developed in Birmingham. And we are fortunate because we were able to partner with them. And they provided us not only with the initial data, but they've also done a second validation study, which allowed us to do really accurate what we believe to be accurate sample size calculations. So we feel we're in good shape from that perspective. But the SBQ is the primary outcome measure.
Great, thank you. You mentioned CD8 exhaustion, how long the CD8 cells were exhausted, and was it temporary or permanent in persistent long COVID?
Yeah, I can't answer that. It's consistent with what other groups have seen, so we know it's there. As you saw, we had blood samples from 3 months all the way out to 4.5 years. We've not done a full-time analysis on the actual cellular deconvolution. To be frank, the cellular deconvolution is useful using proteomics, but it would be much more useful to do direct measurements.
Thank you. And another one here, how do your results align with Jeremy Nicholson at Australian National Phenome Lab?
I'm sorry, I would have to take a closer look to answer that accurately. Just off the top of my head, I'm sorry, I can't give you a great answer.
Okay, thanks. And another question about, are you still taking participants or is the study full?
So the clinical trial is starting next month, and there are sites in Canada, US, South America, Africa, and Italy. So it depends where you are. The primary site will be Sherbrooke, which has a large patient population in Canada.
And just to let you know that we do at Long COVID Web are happy to host any recruitment materials that you have if you have ethics approval to recruit through a website such as ours that we can share with our patient populations. Yeah. So,
great.
Good. And I think we have time for a couple more. Anne Bear has asked again about your primary outcome tool. I think, has it been validated for responsiveness?
Yeah, so it's been validated now in 2 studies, one of which is public and one of which will be out shortly.
That's terrific. Good, thank you. And maybe you could tell us a little bit about the timeline for all of the clinical trials that you're launching there.
So we chose the specific sites based on the number of patients they were currently seeing, what their waiting list was. So if all goes well, phase 1 will be completed by December. And then we'll probably need a few months to do the interim analyses, and then we'll determine where to go from there. We do have ideas for other add-on medications and other standalone drugs should this not be satisfactory, or if the safety profile, for example, is inadequate.
Okay, that's terrific. Oh, what about children? Has this been considered in terms of recruitment or participation?
So I'm a pediatrician, and I can tell you it'd be very, very difficult to use anti-inflammatory drugs, which are this powerful in children, to start off with them. So we pretty much have to start off with adults. I just don't think we'd be able to get children enrolled yet. If this turns out to be positive, then we'd have to do a full safety review and see what can be done from that perspective.
Fantastic. I think we'll call it a wrap there. It's 2:00 and Eastern time. And I just want to say a huge thank you to our two presenters. Somebody said it better than I. Awesome presentations. Thank you very much. Thank you everyone for your questions. And we do have the survey posted in the chat there. If you have a few minutes just to give us some feedback, that would be terrific. So thanks everyone for joining us. We'll be back in May and hope to see you there. Thanks so much. Bye-bye now.
Thank you.